All right, welcome everyone. We are here today to talk about Roman fever, paludism, tertian or quartan fever, ague, marsh fever, Vishama Jwara, Dumada, Malungo, malaria. So this is an ancient disease. As you would suspect by its various names, it's characterized most notably by cyclical fevers. And it's been known by diverse names throughout human history in the geographically disparate places where it occurs. And it's an old disease. Given the tens of thousands of years we've been living with malaria of some form, it's surely been known by many more names that have long since been forgotten in languages that have gone extinct. Thanks everyone for coming. My name is Dyann Wirth. I'm an associate professor of immunology and infectious diseases at the Harvard Chan School of Public Health. The importance of malaria to our species and to global public health is reflected in the diverse research that's focused on this disease at Harvard, in lots of corners of Harvard. And in today's symposium, we're going to hear just a sampling of this research with a focus aimed on work that's aiming to combat this disease. So we're also going to be fortunate to hear from our special guest, Dr. Fitsum Tadessa, who is a world-leading malariologist from the Amrar Hansen Institute in Ethiopia, on the current state of malaria in the Horn of Africa. So this symposium today, entitled Malaria in a Changing World, is a triptych. It's a symposium in 3 acts with 3 talks each. So the first act, Past Lessons, is going to explore our species' history with this disease, from acquisition of malaria as a human disease to its spread around the world and humanity's efforts to control it. The second act, Present Challenges, will feature 3 talks that will explore current hurdles in today's world for malaria control in different geographies. And the third act, Future Solutions, will highlight innovative work that's happening at Harvard and elsewhere, and that will hopefully inspire optimism that someday the curtain will close on this disease. So before we get started, I just want to briefly thank the sponsors and co-sponsors of this symposium, the Harvard Global Health Institute, the Harvard Defeating Malaria Initiative, the Harvard Undergraduate Pre-Med Society, and the Nigerian Students Association. Just a note on format, we're not going to have Q&A after each individual talk. We're going to have Q&A at the end of each act. So after 3 talks, we'll have all our speakers back up here to sit in these chairs, and we will have questions moderated by representatives of the Nigerian Students Association. And everyone has a mic, so everyone is encouraged to ask your questions, okay? All right, with that, I think we're ready to get started. And our first speaker today is Dr. Manoj Duraisingh, the John Laporte Given Professor of Immunology and Infectious Diseases at the Harvard Chan School of Public Health, who's going to tell us about the origin story of malaria.
Origin story, my goodness. All right, Act 1, Dan, thank you. Oh, great to see all of you, great to see a full room, very exciting. So, yes, I was challenged with this by Dan, so to be a little bit broad at the beginning, because, just to set the scene with malaria, but then to go deep into an origin story. So here's here's me trying to be provocative, and, if that can be debated in the breaks. What is amazing is this, that, 100 million people have died. And so that's a lot of people. But, more sobering folks think that the actual number is that, which is still a lot more than, tuberculosis and other major pathogens of today. And this is all just to say that, not only is it an ancient human disease, it's been a pretty lethal one. The burden is global, but very disproportionate at the moment. And if you take a century kind of long view, you see this. The burden was really a lot greater at the turn of the last century. And depending on what kind of person you are, yeah, the folks at HSPH kind of fall into two categories generally. A lot has actually been achieved in that century, but we really need to finish the game because things have not changed much in the last 10 years. We now live in a malaria-free country largely. Just a little point is that a huge amount of effort went into malaria elimination, leading eventually to the CDC, as we know it. So, the first question was, what makes me ill? Dan was very eloquent a second ago. Malaria, swamps. It took a while, but this gentleman here, Laverin, actually saw active forms. He actually attributed the disease to these forms, which are actually the transmissible form. It's credited with spearing mosquitoes, but actually that wasn't the infection that was associated with the disease. Sorry, he wasn't the person who discovered the mosquitoes. It was actually this person who discovered that mosquitoes transmit Plasmodium parasites and saw oocysts on the midgut of some mosquitoes. And the importance is that not only do you associate the parasite with disease, you now know how it actually gets between people and is a pernicious vector-borne disease. And I've always wanted to stand in front of a giant mosquito, and so here we are. That wasn't the end of the story. I mean, there's actually a liver stage, and this is very important, in terms of elimination and eradication. It took a lot longer to actually identify that because there are very few parasites in every liver. It ends up with the life cycle that we know. The microscopes got better. You could first see red cells. You could then see these lively forms in the blood. Then the midgut of the mosquito. And finally, the liver stage, which gives you the life cycle that we know, with all clinical disease being associated with the blood stage. So Dan actually asked me to focus on falciparum, so here we are. But there are lots of Plasmodium that can infect humans. They actually form 4 subgroups. There's falciparum, there's the vivax clade, malaria, and also ovale. You can see all of those here. And only these are really found to infect humans, and only a few really show human-to-human transmission. So the question then is, where did it all come from? We're supposed to talk about the deep history today. This is probably embedded in everyone's brains at the moment, but that's not the answer, as we know. But we have all suffered from this. It shows the importance of knowing the origins of pathogens that cross over. Another approach that our lab is quite interested in is just taking a comparative evolution approach. And these are a whole bunch of organisms that are related. Believe it or not, Plasmodium is related to Paramecium, quite distantly, but related nonetheless. And what happened was that an organism that liked feeding on things by ensnaring them essentially realized it's a good idea to get closer and closer to a cell. And so that's what's happening here. This is cellular vampirism. Cryptosporidium, another human pathogen, gets a bit closer. Toxoplasma and Plasmodium go into the cell. And babesiosis, our local malaria, actually gets in and then gets out again to feed really effectively. And so you have this picture. Most of these pathogens actually invade different kinds of cells. But this is the deep evolution view. I think the real danger now is how would a new Plasmodium parasite get into the human population now. And so some really great work by Dr. Beatrice Hahn, has blown this field open. So here you can see 2 parasites. They look identical. And they actually are identical in this case. But the point is that this could be from a human infection, and this could be from an ape infection. This has been known for about a century. So what it really required was sampling of of lots of gorillas and lots of chimps and lots of apes to actually understand the relationship between human and ape parasites. And this was done actually by getting fresh feces samples and utilizing molecular analysis by PCR. And this was what they found, which was fascinating. There are actually a whole bunch of related Plasmodium species in apes, in the great apes, that are related to Plasmodium falciparum. But look here. Plasmodium falciparum is nested within all of these pre-falciparum isolates, which implies that at some point, an ancestor of pre-falciparum emerged into the human population. And in fact, that's true with all existing falciparum. And so someone told me that this took a lot of energy to generate. It's 4,002 joules in total, which essentially is to make the point that falciparum came into humans from gorillas. Now, gorillas, you can see here, the blue guys, they actually harbor a whole bunch of other Lavranians, and as do chimps as well. They have other Lavranian species. So the question is, how often do they actually jump? So before we get to that point, when did falciparum enter? And I hope we hear a little bit more about the more recent history in the next talk. I'm looking forward to that. But these Western lowland gorillas are located here, and the idea is that this is when they actually jumped over. This is still uncertain. It's being hotly debated, but the idea is that that event only really has happened once. And so there are lots of barriers, and they could be molecular, ecological, which actually have prevented it from happening over and over again, which can lead, as we know, to deadly pandemics. And so understanding those barriers is a large goal of many laboratories, including our own. So how did it jump? We take a molecular and cellular view. And here is a little parasite, which, isn't going to move. But that parasite can attach to a red cell and get into it via its nose. And the first thing that it sees in a new host, so it's going from a gorilla and it's getting into a human host, would first be the liver, but then when it gets into the bloodstream, it sees the surface of the red blood cell. The red blood cell surface, turns out, is the most polymorphic surface in the entire human body. We know this because of transfusion biology and blood groups. Lots of the proteins are variable, but also the sugars on those proteins. And so here, one of the first major variations between humans and apes. All right, some of you recognize our professor. It took a little while, but okay. So are the sugars that are the terminal sugar on these elaborate chains on these proteins. That's called sialic acid. And amazingly, humans and New World monkeys have a very different form to apes and macaques. And it's just a small change, but it has a huge effect on lots and lots of pathogens, including flu and many other scourges of humankind. So a PhD student, now a professor at UMass Amherst, Selassie, did a great experiment. She basically changed the sugar on these red blood cells, these human red blood cells, to the ape and the macaque-like variants. And she just did this by genetics. And that's what our lab is. We're a genetics lab, so that's what we like to do. And so the question was, okay, if we change that sugar, so here's the human, and this is showing that the ape sugar variant is now on the surface of those cells, what happens to those parasites in terms of being able to get in? And so here's a parasite getting into a cell. And as it happens with falciparum, which already can kind of get into human cells, not a lot, but Knowles-I, which normally invades macaque cells, which has the ape-like sugar, suddenly gets in really, really well. So this is a real restriction factor that parasites have to get over to actually enter into the human population. And so that's what's written down here. And it's a simple deletion in a gene that has happened in humans. It's happened in very few mammals, but it has happened in a few. It's happened in ferrets and, as I showed you, New World monkeys. And strangely enough, it's suppressed in dogs who are kind of quite close to humans. So a lot of work has now gone into thinking about those parasite proteins, these kind of keys that are cognate to the locks on the red blood cell surface and how you actually get in, because we want to stop the parasites from getting in. This is what we now know. There's one parasite key for one that should be a host receptor. They're all kind of polymorphic. The keys are polymorphic. They can sometimes be used and sometimes not. They can be expressed variantly, but they can lead to differences in their ability, this variant polymorphism and expression in terms of what receptors they actually use. I showed you something about sialic acid. Most of these receptors here are the main carriers of sialic acid. If you look over here, it's quite interesting. There are a few of these protein receptors. And one of this is actually a protein called basogen. And basogen is actually quite fascinating because the parasite key, the protein that it uses, RH5, was actually identified. But what was then found was that it actually transferred from one of those ape parasites to another ape parasite to produce a new line. This is prefalciparum, that ancestor, which essentially was a required change before that tropism, that shift from from the gorilla to the human actually happened. So RH5 was very implicated in that. And so polymorphism in the RH5 protein has become a major determinant of human tropism. So that's interesting from the tropism point of view. But a second fact is fascinating when you tie it together with that point. So again, taking that red cell genetic point of view, now another PhD student, Amy, and this time now a professor at Yale University, she did something else. She took away one of those glycophorins, the things that sialic acid, that has sialic acid on it. And some parasites care about that, and some just don't. But when you take away basogen, it turns out all parasites actually need that receptor to get in. It's a strain transcendent receptor. So it looks like the key protein that was needed for the parasite to move from into the human population is also essential for all parasite strains today to actually get into human red blood cells. So RH5, the ligand that binds to that receptor, is now a major blood stage vaccine candidate. We're now living in a vaccine age for sure. Very exciting work with liver stage vaccines, but also with blood stage vaccines, the work of Simon Draper and lots of his colleagues. And recently, they're actually finding that when you vaccinate with RH5, you actually can get very, very good protection against infection. So a goal of the lab is to identify other parasite determinants of tropism that we believe could be essential for invasion. I need to show this before Dan kicks me off the stage. How many of you know about Little Joe? Little Joe's been a part of the life of our lab for many, many years now. Little Joe's leaving Boston. It turns out he's leaving Boston for a good reason. He's required elsewhere in Pittsburgh, it turns out. So how do you actually map those new determinants? And I'm not going to tell you much about this at all, but it turns out that extant P. falciparum strains can invade gorilla cells versus human cells to different degrees. And we can do 4 genetic mapping experiments. So that's what we do in the lab. And this is just the peak of what we found. There are actually a bunch of loci that are associated with differential invasion into gorilla versus human red blood cells. And these are the folks in the lab doing it and our collaborators. And just to finish, it's an ancient scourge. It's jumped from Western lowland gorillas into humans. There are sugar determinants and protein determinants, but falciparum found a way to invade the entire human population. Why is this important? For surveillance. So far, there has been no evidence when you look at human populations neighboring gorilla and chimp populations of the other ape-like, falciparum-like parasites crossing over. And then we're trying to identify the molecular determinants because we believe that's a way of informing vaccine development because all existing falciparum parasites may require them for invasion. These are the folks, most important people, and I'll stop there. Thanks.
All right, thank you, Manoj. This is your phone. Our next speaker is Dr. Megan Michel, who is a postdoctoral fellow in the Department of Human Evolutionary Biology, who's going to tell us about the next chapter in malaria's history. All right.
Hi, everyone. First of all, thanks to the organizers for the invitation to come and talk to you today about some work that we did previously using ancient DNA to try to explore the history of human malaria. And I'm here today presenting some work that was led in part by Dr. Christina Werner in the anthropology department, who unfortunately couldn't be here, but I'm glad to be here instead. So we've just heard a lot about the origins of Plasmodium falciparum malaria, and I'd like now to zoom forward a bit in time to malaria's more recent history. And we can see from this map that only 100 years ago, the landscape of malaria infection was much different to how it is today. So in the year 1900, malaria covered 50% of the world's land surface, reaching all the way up into the southern reaches of Canada and Siberia. And it's thanks in part to a century's worth of public health efforts and developments in infrastructure and hygiene that we now have a much smaller region where malaria is endemic today. So in my work, I'm interested in using ancient DNA to learn more about how malaria attained this global distribution, look at population affinities of parasites from these regions where malaria is now eradicated, and ultimately to try to understand something about the processes that gave rise to parasite populations that still impact people today. So as anthropologists and archaeologists, we have multiple different sources that we can turn to try to learn about malaria's history in human populations. We know about references to malaria in ancient texts from the Greeks and Romans, for instance, who describe cyclical fevers, as we've heard before. Osteologists also have spent a great deal of effort trying to look at markers of anemia and the distribution of these anemia-associated skeletal markers to try to understand where malaria was endemic in the past. But it's really only been within the past decade or so that advances in ancient DNA technology have enabled us to isolate genome-wide data from ancient humans that lived thousands or even 10,000s of years ago. And really importantly for us today, we can also sometimes isolate genome-wide data from bacteria, viruses, and eukaryotic pathogens that infected those people at the time of death. This is a really exciting time to be working in this field because ancient DNA work on malaria is relatively new. There were several partial mitochondrial genomes from Plasmodium falciparum parasites isolated from some people in the Roman Empire, and also some really important work on these old blood slides, one of which you see pictured here, about 100 years old, giving us some first glimpses into the population affinities of European Plasmodium vivax and falciparum in regions where it's now eradicated. But partially due to the biology and genomics of these parasites and also characteristics of working with ancient DNA in general, this is working with ancient malaria DNA is a rather difficult problem. This is partially because these molecules that we're working with are damaged and highly fragmented. So as soon as an organism dies, the DNA begins to break down, and we're usually left with molecules that are between 30 and 70 base pairs long. So they're really hard to map often. Even more importantly for us, ancient microbial DNA is typically present in extremely small quantities within an ancient human bone sample. So in this little cartoon, the majority of DNA comes from environmental contaminants, things like soil microbes that can look quite similar at times to organisms that we're interested in studying. A smaller amount may be from humans, and a tiny fraction of a percent will be from the parasites that we're actually interested in looking at further. So this makes finding these samples kind of like looking for a needle in a haystack. Fortunately, we've developed laboratory techniques to help us deal with this problem, in particular hybridization capture, which is a technique where we use synthetic DNA baits, which are designed to be complementary to the genome we want to study, to actually physically bind to and fish out the DNA molecules from the parasites that we're interested in. So for my PhD work, I designed 2 capture arrays targeting the mitochondrial genomes of 5 human-infecting Plasmodium species, as well as the nuclear capture arrays of Falciparum and Vivax. A second major development in the field of ancient DNA that paved the way for this work is just the really large increase in the number of ancient individuals that have been studied using these methods. So for my PhD, I was fortunate to work at the Max Planck Institute for Evolutionary Anthropology in Germany, where they've collected and studied thousands of ancient individuals, mostly to learn about human population genetics. But for me, this was a really largely untapped resource for looking for these rare cases where we could identify preserved malaria DNA and skeletal material from these individuals. So taking advantage of this collection, we were able to extract Plasmodium DNA from 36 ancient individuals, including cases of Vivax falciparum, Malariae, and several individuals who are co-infected with more than one species. And we were really excited by the fact that these span quite a range of the human past, about 6,000 years. Particularly for Vivax, we have cases from across Europe going back about 6,000 years ago, so begins to tell us something about the distribution of these parasites well before the development of written records. But for the remainder of this talk, I'd like to delve a bit deeper into a couple of case studies. And when we started looking for patterns in this data set, one thing that emerged is this link between human mobility at multiple scales and the spread of parasites throughout the world. So I'm going to talk briefly about the spread of malaria at a global scale with a particular strain from ancient Peru, a more regional scale, and finally, a kind of really interesting individual case of a person who we think was mobile, and we can identify their mobility from their infection. So diving right in, one of the things that we were really interested in addressing with this data was the origin of malaria in the Americas. And people have put forward several alternative hypotheses to try to explain how malaria arrived in the Americas, whether Falciparum and Vivax may have arrived during the colonial era, either through European colonizers themselves or as a result of the transatlantic slave trade, whether particularly Vivax, which is generally more cold tolerant, might have arrived with the first indigenous inhabitants of the Americas, or whether it might have spread via contacts with Oceanian seafarers that potentially predated European contact. So we were fortunate to recover some relatively good quality data from an ancient individual from this site here, Laguna de los Condores, in what's now Peru. And this individual was associated with the Chachapoyas culture and dated right around to the period of European contact. But through the archaeology alone, we weren't able to say whether they pre- or post-dated the contact period. So using the really rich set of modern Plasmodium vivax genomes that have been generated from around the world, we wanted to put this ancient lineage into that diversity. So here I'm showing a principal components analysis. You can think of this as a sort of genetic map, with each point representing a modern genome, and points that are together in PCA space are more closely related genetically. So here we can see that the x-axis separates out populations from Africa and Latin America from Southeast Asian and Oceanian populations over here. And if we project our ancient Peruvian strain onto this diversity, we observe that it actually falls kind of right on top of the cluster of modern parasites that exist in Latin America today, which is consistent with what we might expect and suggests some level of population continuity since the early contact period. We delved into this further statistically, and although I won't kind of dive into this in detail, we were able to show that the Laguna de los Condores strain was more closely related to parasites that live today in Peru, as opposed to strains that are endemic in other countries across Central and South America, again, kind of pointing to at least some level of genetic continuity in that region. And really interestingly, when we then overlay the diversity of ancient European strains that we recovered on top of that, we found that they also cluster quite closely together with the ancient and modern Latin American lineages. So we think this provides relatively strong evidence that Latin American Plasmodium vivax likely resulted via transmission from European colonizers during the post-contact era. This is also kind of supported through phylogenetic analysis, which placed the European strains basal to modern and ancient Latin American Plasmodium vivax lineages. So in the interest of time, I'm going to skip some of the analyses we did on Plasmodium falciparum, but I wanted to point out that we recover a rather different history for that species in the Americas compared to Plasmodium vivax. And it's already known from kind of modern lineages, which are really well studied, that the African Plasmodium falciparum strains are really closely related to strains present in South America today. Unfortunately, we don't have any high coverage falciparum European genomes other than those blood slides that I mentioned to you earlier, which are right here, this EPRO 1944. We can already see that they look rather different from the African and South American strains. So we think this is probably more parsimoniously explained via transmission of falciparum from Africa as a result of the transatlantic slave trade, with vivax instead probably coming from Europe itself. So zooming into a kind of interesting, more regional case study, we found several malaria infections from a site in Belgium, a cathedral cemetery from the city of Mechelen. This is a map of the cemetery. It's absolutely like full of human graves, over 3,000 that have been excavated so far. And what's really interesting about this site is that it's situated directly next to a Spanish military hospital, which was constructed by the Habsburg Empire in the 1500s to support the wars that they were conducting in the Netherlands and Belgium at that time. From the archaeology, we knew that there were kind of two transects of burials at this site, so an earlier time phase from about 1100 to 1400 CE, and a later phase from 1400 to 1800. We know that the hospital was active during this later phase, and we found a difference in the human genetics of the individuals that were interred in these two layers of the cemetery. The earlier phase was about half male, half female, and genetically their ancestry looked how we might expect modern Belgian populations to look. In this later phase, we found almost exclusively male individuals, and their genetics was much more diverse, with elevated Southern European ancestry, likely from the Mediterranean region. Interestingly, we also found malaria in some of these individuals. In this earlier phase, we recovered 2 cases of Plasmodium vivax, whereas the later phase had 8 infected individuals, including infections with falciparum, vivax, and malariae, as well as a couple of co-infections. And what was kind of interesting to us is that it's not thought that falciparum was likely endemic north of the Alps during this period, since it requires higher temperatures to sustain ongoing transmission. So sort of looking for ways to explain this, we learned that the Habsburg Army of Flanders actually recruited soldiers from the southern Mediterranean, from northern Spain and Italy, that then traveled to Belgium and the Netherlands. So we hypothesized that these all-male individuals with this more Mediterranean ancestry may have traveled from more southern climates, bringing their parasites with them. And that could explain why they were buried in this cathedral cemetery in Belgium. So the last case that I want to mention to you is an individual from the site of Chakapani in Nepal, about 3,000 years old. This is one of the most interesting individuals in the study for me, mostly because I was convinced it was a false positive for a really long time, because it's not a site where I would have expected to find malaria. It's a high altitude site located at about 2,800 meters above sea level. There are these cliffside tombs. The individuals were interred there, and it's pretty cold and quite dry, so not an environment that would be somewhere we would think of Plasmodium falciparum undergoing transmission. So we were able to reconstruct a full mitochondrial genome from Plasmodium falciparum, as well as nuclear data. So we started looking for explanations for this pattern that we were seeing. And talking to the archaeologist, we learned that the site of Chakapani is situated in a transverse valley that was a trade corridor connecting the Tibetan Plateau to the southern reaches of Nepal and the Indian Terai, where malaria is endemic today. There were also artifacts at the site that suggest links with northern India. So we think that it's possible this individual could have been a trader or someone who traveled from high elevation to low elevation during their lifetime, where they contracted the illness before returning to high altitude and dying. So to conclude, I think I'm really excited about this work because I think ancient DNA is beginning to provide an opportunity for us to look back into the past to try to connect what we're seeing in terms of pathogen genomics and malaria genomics with the human host as well, looking at ancestry and potentially changes in natural selection as well, and environmental shifts. So really trying to connect all of these factors that shape risk of malaria in the past to hopefully understand how changes that are continuing to go on today impact malaria spread and endemicity. So with that, I'd like to thank all my co-authors and all of the funding sources that contributed to this work.
Thank you so much, Megan. So for most of human history and our association with malaria, individual people and societies just suffered with this disease. But in more recent centuries, we've been fighting it. So to hear about the recent history of malaria control and treatment and prevention, we are going to hear from Dr. Dyann Wirth, who is the Richard Pearson Strong Professor of Infectious Disease at the Harvard Chan School of Public Health, and who is also the chair of the Malaria Policy and Advisory Group at the World Health Organization. Thanks, Dan.
Great. Thanks, Dan, for the invitation. And indeed, it's a pleasure to be here and see this large group of people interested in malaria. So now I'm going to jump ahead to more or less today, or last December, actually, and talk to you about the situation as we speak today of malaria, and then kind of reflect the last, after these talks, the very recent past from the 1960s until now. And the 1960s are important because that's the first time there was a worldwide effort to eradicate the disease using, at that time, innovative tools. And that will be part of the theme of what I'm going to talk about is innovation. So this is from the most recent World Malaria Report, which was issued last December and reflects data primarily through years 2024. And it's important to realize that as malaria remains a critical and important public health problem today, we are making progress. That is, the number of cases and deaths averted continues to increase, and intervention measures, prevention and treatment in particular, are preventing these cases. However, of course, malaria remains a serious challenge, and progress that we had been seeing, say, from the early 2000s to maybe 2010 or 2012, has really plateaued. And so this doesn't mean we're not being effective, but populations are growing and we're plateauing in how many cases and deaths there are due to malaria each year. So we need new solutions, and looking back at old solutions maybe can tell us something. This is from the Global Technical Strategy of WHO, the prediction in the greenish line of where we were supposed to be with a great reduction, about a 90% reduction in malaria by 2030, based on predictions that were made in the early 2000s, 2010, 2012. And so we're clearly off course. The blue line is where we are, and at current rates, we are going to not meet the global technical strategy goals. So we're going to think about that. You're going to hear many talks today about some of the challenges. I'll mention a few of them, but it's important to realize where we are. So now let's look back. And this is an interesting graph, which I admit helped both from ChatGPT and BioRender. So I take some responsibility, but I did have help. If we look at the number of countries certified malaria-free as sort of a surrogate for how successful malaria control has been, we see an interesting pattern. There was actually quite a bit of certification in the 60s and 70s, including countries in Europe and the Northern Hemisphere. But then it just plateaued. There was really nothing, or so little, hardly worth mentioning, for almost 40 years. And then recently we've seen a resurgence in certification, that is, and an emphasis on why, and we're going to talk about that, an emphasis on malaria elimination in countries and ideally worldwide eradication. So in the 60s, there were two innovative tools: chloroquine to treat the infection, and DDT to be able to kill mosquitoes. And that was used primarily, although not exclusively, as residual spraying on house walls. And so that's the innovation that led the World Health Organization to call for malaria eradication in the late 1950s, and that led to this activity in the 60s and 70s, was abandoned because I think political will was lost. It was successful in some places, but in the places where malaria is highly transmitted, some you've heard about today, there was really very little progress using these tools. So what's happened more recently? Well, there's the basic discoveries that are actually fueling the current effort to eliminate and eradicate malaria. That declaration having been made now by a private foundation, the Bill and Melinda Gates Foundation, Bill and Melinda themselves at the time, both part of the foundation, declaring that no child should die of malaria. Okay, so these tools were all developed in the 1970s. Up in the right-hand corner is artemisinin. That's 2UU, who won the Nobel Prize for the discovery of artemisinin, the now treatment of malaria, over 400 million doses given each year. Janet Hemingway for insecticide-treated bed nets, the major tool for reduction and what has led to the progress in reducing malaria primarily. The malaria vaccine, which we've yet to see an impact, but it is finally available and in people. And then the diagnostic, the histidine-rich protein discovered by Bill Traeger, and is the basis, or has been the basis, of rapid diagnostic tests. And Fitzen will tell you that that's also under pressure. Okay. So now, again, I chat GPT and biorendering. I asked, what happened between 90s and the year 2024? Why do we see all of this activity leading to elimination in certification and elimination in several countries, and the reduction overall in malaria burden worldwide? And so there's lots of things. There's governance changes, there's funding, there's the Global Fund, the President's Malaria Initiative, the Global Technical Strategy. All of these things drove to malaria elimination, calling for malaria elimination based on the technologies of insecticide-treated bed nets, artemisinin combination therapy, and rapid diagnostic tests. I mean, and they were introduced in the early 1990s, became widespread early 2000s and our tools today. And I think it's important. It was the innovation in discovery and then the governance, the sort of decision by policymakers driven by a desire to give their populations the ability to be free of malaria that actually drove all of this. And we, of course, are now at another crossroads because these tools, great as they are failing, as did chloroquine and DDT in the previous era. And so there's resistance to pyrethroids, insecticides, artemisinin resistance. You're going to hear about these. And there are deletions of the protein that is the basis of the RDT diagnostic. So kind of the perfect storm. And so I think we face kind of several challenges. There are the biological challenges. This is an organism that is incredibly efficient in transmission. And the mosquito and the parasite are incredibly resilient. It's very hard to interrupt transmission over the long term. It has a high reproductive rate. In many settings, one infection can lead to 100 new cases. And you see that in places that have had great reductions. This is from an article now several years ago by Joe Cohen, showing this rebound effect. If you take away interventions, all of a sudden the infection rate and burden goes way up. And evolution is continuous. Manoj talked about this, the evolution of jumping to people, but within the organisms, both the mosquito and the parasite, there's response to insecticides and drugs, and it's an ever-changing target that needs a dynamic response, and that's the challenge. And then, of course, all of this costs money, right? So one of the things that has really fueled the current renaissance in malaria elimination has been the incredible investments by external sources, the Global Fund, the President's Malaria Initiative from the United States, and many other bilateral donors. However, that's plateauing, kind of like the disease. And in the current environment, we all know that at least from the United States, but also from many other countries, the investment in foreign health is somewhat limited, or is being limited. And so we are facing a crisis. I think there has been a call with the Yaoundé Declaration, which was one of the last things on that massive diagram, where the heads of state, the ministers of health from 11 of the most endemic countries in Africa signed a declaration committing to domestic investment. And we see that in many countries, including Nigeria, where there is a significant change in the investment strategy for malaria. So, I mean, we are seeing progress, but it is a challenge. Okay, so let's see where I am on time. Okay, I guess 4 minutes. Okay, so now 4 minutes, I'm going to run through. You're going to hear details about some of these things, but I kind of didn't want to leave you with like the sad note that everything's going down. There's lots of really interesting developments that make me optimistic that we've got malaria on the run. Okay, maybe not as fast. We're not doing as well as we were, but it's going to get better. So there's the vaccine, and this is the Director General of WHO himself is a malariologist, and he was delighted that finally, after 40 years, a vaccine was approved, and now we have a second one approved in a time span of less than 2 years. And so the rollout, there are 24 countries in Africa that have introduced the vaccine, most of them subnationally in areas of the highest transmission where the vaccine is likely to have the greatest impact. the problem of insecticide resistance, I'm sure Flaminia is going to talk about. There are dual active nets, and these now are the majority of nets being purchased by countries, either using their own funds or using funds from donations. And these are effective against pyrethroid-resistant mosquitoes. There are oral drugs, insecticides, that actually the human takes, and it kills the mosquito. Is that going to work? I don't know, but very interesting clinical trial data or field-based trial data showing effectiveness. There's genetically modified mosquitoes. This is a major investment of one of the big donor agencies, the Gates Foundation, and others, to modify the mosquitoes, either to reduce mosquito populations or to change their susceptibility. Flaminia is going to talk about killing parasites in mosquitoes rather than killing the mosquito, another new idea. There are monoclonal antibodies. Dan's been involved in some of this work that are directed against the same protein that the malaria vaccines are directed against. And it's kind of like delivering the end product of a vaccine, the monoclonal antibody. Monoclonal antibodies have been incredibly effective in many diseases, primarily cancers. And so this is an opportunity, perhaps, challenges, but there are always challenges. There are new drugs. This is the latest drug and is now finished. This is CAF 156 or ganaplacide, which was developed by Novartis, MMV. Didier Leroy is here and was involved in that work. It is combined with lumefantrine, and the Kaluma phase 3 clinical trials finished sometime last year, reported at ASTMH to be effective, and this is now before regulatory approval. The Medicines for Malaria venture has invested now heavily in chemoprevention and using the idea based on the successful chemoprevention in HIV drugs, is working to develop a long-acting injectable, a drug that could be injected and would be active for 3 to 6 months, preventing malaria infection. Elizabeth Winsler leads a group that my lab is involved in called the Malaria Drug Accelerator, really finding entirely new targets. If you think about it, almost all the malaria drugs that we are in use today hit a couple of targets. I mean, it's amazing that an organism that has 5,000 genes, 30% of which are essential, there are only drugs to a couple of targets that are actually in use. So this is very important. And she's also developed a model for how to do this involving groups from across the world, including those in Africa. And this is, of course, a very important theme, the necessity to develop these kinds of capabilities in countries that actually have the disease. But innovation is producing more products and faster, but that's, of course, a challenge. Look at this. From 1940 to 2020, there were 1 to 2 products a decade. We anticipate in the next decade 4 to 5 new drugs, next generation vaccines, GMM mosquitoes, new insecticides. So we have to rethink regulatory new regulatory practices. We have to be more nimble. It's also clear there is no magic bullet. I mean, combining interventions is the strategy that has worked. I'd love there to be a magic bullet, but let's be realistic. And finally, the finite funding is finite, so we have to make hard choices. I won't go into those. And then just to finally mention, I know Noor is online, that it's going to be very important to, it's not a one size fits all. And we've just finished a arduous exercise at the Malaria Policy Advisory Group in looking at whether it was possible to find one size fits all. Each area, each transmission zone is different. This is just, I think, an example. I wonder where this is. This might be Kenya. Could it be Kenya? Sierra Leone. Okay. So this is just mapping the different epidemiological and ecological features that occur throughout a country, and tailoring interventions to each of those. So subnational tailoring, major effort, but really enormous progress. You're going to need new ways of measuring the impact of interventions. And this, I personally believe, is going to involve using genetics and genetic markers. I won't spend time on it because I'm getting the sign that I'm done. But we need to rethink. And I guess one of the nice things about having this kind of symposium is there is the opportunity to rethink how we approach things. And it's going to take all of us with our different procedures to defeat malaria. With that, I'll stop. Thank you.
Yep. I'll ask you to take a seat here, and we will welcome Manoj and Megan back up to the front.
Okay.
So we have time now for group question and answer. And I believe that's going to be moderated by representatives of the Nigerian Students Association. So I'm going to give up the mic here and let you drive. But yeah, hi, please introduce yourself. Sorry.
Hi everyone, my name is Frederick. I'm part of the Nigerian Students Association, and I'm going to moderate the Q&A session. So I think because of brevity of time, I would want to first start taking the questions from the floor based on the presentations we've had so far. And then when we are done with that, maybe I'll add a couple of questions. So if you have any question, you can just wave your hand and then I'll call you. All right. Anika? Sure.
I'm curious, also having a background in evolutionary biology, like Megan, I'm curious what the kind of biological and evolutionary history of malaria can tell us about the effectiveness of potential future prevention methods and treatment methods, and perhaps maybe where the disease itself is going and what we should be anticipating in the future.
That's a good question.
Okay. Yeah, that's a great question. I think that currently, like the insights that we can get from ancient DNA about malaria are more are not so practically useful yet. My hope is that I think sort of one of the main challenges is kind of the low recovery rate that we observed like in our study. So I looked at metagenomic data from thousands of ancient individuals. We found only a handful, but from the estimates that we've seen about the death toll of malaria over time, we know that it was much, much more prevalent. So I think we have to be a bit creative in how we use the anthropological data and the paleogenetic data, maybe looking into human markers of adaptation to malaria to understand sort of more about how the host and pathogen have evolved together over time, maybe a bit more creative sampling. So instead of sort of taking advantage of what was already kind of there, which I think made sense at the time, like more targeted sampling of regions where we expect better preservation or, those old blood slides sort of to try to get a sample where we have enough information to look at the evolution of the parasite itself rather than global spread.
Yeah, I mean, I would say, there's the classic markers, right? I mean, there's the sickle hemoglobin, which is protective to humans, right? And so presumably there are similar markers on the parasite for having been in humans and maybe having been selected by humans. The drug resistant markers are obvious. You treat with a drug, the parasite which is resistant will grow out. Okay, so there are clearly footprints, and I think you ask an important question whether we have the resolution to see that. I don't know. I think that's the question, and certainly there are on parasites regions of selection. We can demonstrate regions that are selected or appear to be under selection where we don't know the cause, and perhaps that is an effect of humans or something else that we haven't yet measured. So it's a great question. Don't know the answer.
Okay, thanks, Manoj.
It is. So I wanted to call my talk Back to the Future, but it didn't seem to work. But maybe that answers your question a tiny bit. But here are two interesting points that I think you can get out of looking backwards. One is very practical. In many fields, they do what's called ancestral reconstructions of antigens. And that's a small thing that's maybe worth doing. It's been done in other fields. But here's a plug. Dan asked me to talk about falciparum, but what I actually liked about your talk is that it was largely focused on Vivax, because Vivax was robust and probably will remain, one of the biggest challenges when you get closer to end elimination and eradication, and that might be a small lesson from the past.
All right, thank you very much. Any other questions from the floor? Okay, I can see.
Hi, I'm Bruno from Boston College. This question goes to all of you. From the presentation from Dr. Morgan, you presented that malaria entered to humans basically from African countries. And also, Megan and Dyann, you also mentioned there are some countries that are malaria-free. So I'm asking, why didn't this malaria eradication and elimination, since it's entered through Africa, so why didn't it start from Africa?
It's a question I often ask. Why not start from the hardest places, right? But in fact, it started from the easiest places, and the countries that, in general, are being certified are countries that never had highly endemic, deeply embedded parasites. So that represents the biggest challenge, and I think it was not feasible. In fact, in the eradication campaign of the 1950s, Africa was kind of written off as impossible, and I think does not reflect well on the history, but it's going to be much harder as we see. I mean, I think Fitzum is going to talk about the situation in Ethiopia, and you see this in other places where you see, some effective control, and then because of this high reproductive rate, you get rebound with just a little change in interventions. So it's a critical question. I love that question, and we should start from the hardest places. And I think that is the World Health Organization and Rollback Malaria have just kind of launched this thing called the Big Push, which has its origins in the high burden, high impact program of the Global Malaria Program from a few years ago, with the idea that most of the effort should be located in the place of greatest risk.
All right, thank you. Go ahead, please.
Maybe related with that same question, Dan, and also referring to your last slide on rethinking. So do you think we can reach to that last milestone in these high burden settings only using, for example, biological interventions, or you're rethinking as non-biological interventions? Or is this a general phenomena? Yeah.
I don't think there's going to be a single bullet. Not everyone agrees with me. I mean, there are people who believe that genetically modified mosquitoes will dramatically change, and but my personal history tells me that every time someone thought there was a magic bullet, it wasn't so magic. But it is true that combinations of interventions have made significant impact, and you see that in many places. But in the even I mean I am on the Amen panel for Nigeria, and even their MIS survey was just completed from 2025 compared to 2021. They've seen quite significant reductions in many parts. Lagos State, for example, has relatively little malaria compared to a decade ago. So I think it's going to work. But and then your question is, do you need vector control of vector breeding sites by non-biological means? Do you need other interventions? If you have a great idea, yes. There were laser zappers for mosquitoes. Those didn't work so well, but I think we should let 1,000 ideas blossom, because you never know.
Thank you very much. I can see two hands at the back, so there's a gentleman at the back.
All right, thank you very much. This is Frank from Manoj's lab. Maybe let me stand so that you can see me. So great talk, but my question is directed to Dyann. All these, and I love the fact that you are very optimistic about the progress we are making and new innovations coming up. Like this monoclonal antibodies is something that is also fascinating, right? But I'm thinking about the cost of all these new products that are coming up, because the most vulnerable of these diseases in Africa and Southeast Asia, and we all know that economically these areas are not stable. And so if we are making products that are less or not affordable, then it means that the most vulnerable are not going to truly benefit from that. And so I wanted to know, since you work with WHO, do you have some of these discussions to ensure that the products that are coming up are less expensive and affordable for those who are vulnerable?
Yeah, it's a very important question, but I think there's a paradigm shift happening. You saw it. I don't know how many of you attended the Global Health Symposium a bit earlier this spring, where this paradigm shift of moving to domestic funding, domestic manufacturing, I think is not only an aspiration, but is happening in many places. And I think that's one of the answers. It's always a consideration, but it's the fact that innovation is expensive at the beginning because there's more development cost. You don't have the massive discounts of huge orders. And so it's always, and Didier's here from MMV, it's always a consideration in choosing between new products. But if you set the bar too low, you might not get innovations that are going to actually be game changers. And it's always a tension. And I think one of the things that I see happening is governments facing the reality that the health of their populations is an investment they're going to need to make. In many ways, the global development infrastructure bolstered many of these governments' ability to deliver health, and that money is less than it was previously. And there are more people to treat. So there's a paradigm shift. It's not one I would have chosen or think is necessarily being done in the right way, but it is a fact of life.
All right, thank you very much. Because of time, we will shelve the last question to the end of the presentations. So thank you to our presenters.